Hello class, this is part 4 of chapter 3. We're going to go over proteins as a group of organic molecules. So we have a little bit of vocab with proteins. The first thing we need to talk about is the monomer or the building block, which we call amino acids. So amino acids are the building block, they're the monomer. When we link amino acids together, we get a chain of them called a polypeptide. And the covalent bond that holds our amino acids together is a peptide bond. A protein is one or more polypeptides that have a function. So just because amino acids are linked together into a polypeptide, that doesn't mean they're functional. Polypeptides are special in that they fold into a three-dimensional shape. And they're only functional when they're folded. Sometimes they're only functional when they're folded with other polypeptides. So at whatever level a protein is functional, it can be one polypeptide or multiple polypeptides folded together. It's only called a protein when it's functional. So proteins have many different functions. If there's a job in the cell, a protein probably does it. Many are enzymes, which are involved in metabolism. Remember, metabolism can involve building things or breaking things down. So both directions, but it always involves energy. You tend to use energy to build things and free up energy when you break them down. So enzymes are not just, they don't just degrade, they also build. Some proteins are for support. The most common protein in your body is collagen, and it's just structural. It helps keep your skin attached to the underlying tissues, which makes your skin smooth. Collagen's also, it also gives bones and teeth and hair and nails some resilient strength. So collagen's pretty much everywhere. Some proteins are for transport. They can move things in and out of a cell or in the blood. Some are for defense. They help fight invasions by pathogens. Some are for regulation or communication, really. So they help regulate homeostasis by communicating with different parts of the body. So hormones are chemical messengers, and many of them are proteins. Some proteins are for movement. The part of your muscle that actually does the contraction are based on two different proteins. So some are mechanical like that. So there are many different functions of proteins. One of the things we want you to know is the basic structure of an amino acid. And remember, amino acids are the building blocks of proteins. They are the monomer. There's 20 amino acids that we use. And, there's more out there. We found more than, some weird ones on meteorites. But there's only 20 used by life here on Earth. They all have the same structure, an amino group, which is the NH2, the acid group, the COOH, an R group. This R group stands for anything. This is the difference between the 20 amino acids. All the rest is the same. Let me get rid of that. So when you look at amino acids, everything in blue is the same, and it's the R group that differs between amino acids. The last part is a hydrogen, and all four of these are connected to an alpha carbon. That's the symbol for alpha. So you may need to know what's attached to the alpha carbon. It's an amino group, an acid group, a hydrogen, and an R group, or sometimes that's called a side chain, depending on the book, or who you Google. But know that basic structure. Here's many of the amino acids. I don't think it's all of them. But again, you can notice the amino group on all of them. The acid group on all of them. The hydrogen. The alpha carbon, and in the shaded boxes are the R groups. And you can sort amino acids based on the R group. That's what they've done. So these 5 at the top are nonpolar or hydrophobic. Their R group is. The rest of them is ionic. This group is polar, so it might have an OH or an NH or an SH. And then this group are acidic, so they lose a hydrogen. Well, these are both acidic and basic, because I see arginine in there. You don't need to know them by name. You don't need to recognize each one. Just be able to recognize that these are amino acids. And again, looking at their parts, finding the amino group, the acid group, the hydrogen, and the R group, that's the way to identify that these are amino acids. When we link them together, the acid part of one will link to the amino part of the other. And we'll get a special carbon to nitrogen covalent bond, and that's called a peptide bond. And remember, when we're linking 2 together, that's a dehydration synthesis reaction. If we were breaking them apart, that would be a hydrolysis. Proteins, let me back up. Amino acids, when we link them together, they don't just make these long chains like the carbohydrates. they actually begin to, those R groups interact with each other, and even the amino and acid groups have some electrical interactions, some ionic interactions or hydrogen bonds. And so they start to fold in on each other and get a 3D shape. And there's 4 different levels of protein structure. I want you to be able to describe each level and tell me what bonds are at each level. When they're folded, proteins function, and when they are unfolded, they lose that function. When they unfold, we denature them. So denature is just our science term for unfolding the protein. Alright, so let's look at the 4 levels. The first is the primary level. It is unfolded. So there's no folding going on yet. At this level, we've just linked together our amino acids. It's relatively linear, you know, it might be a little wavy, but it's basically one line. You're not getting any cross reaction with different parts. It's still pretty linear. It's just the order of amino acids, and proteins can differ in the number of amino acids. They can differ in the order of amino acids. And that's how we get different proteins, different polypeptides. And the bonds you can find at this level are just covalent ones. And those are the peptide bonds between the amino acids. So the next level is the secondary level. You get local or partial folding. What I mean by local is parts of the chain close to each other will start to fold together. There's some nice neat keywords for the secondary level because your protein starts folding into these recognizable shapes known as alpha helices. So that's plural for helix. And beta sheets, which if you looked at them on edge, you know, they kind of look like an accordion. But they make these broad sheets that are in proteins. And at the secondary level, you get hydrogen bonds as well as the covalent bonds from the primary level. And hopefully they'll show a picture of the helices and sheets in a minute. Next is the tertiary level. It is fully folded. But one of the keywords for the tertiary level is one polypeptide. And the main difference between tertiary and quaternary is the number of polypeptides. So you get 3D shape, it's fully folded, you get hydrophobic interactions, hydrogen bonds, ionic bonds, and covalent bonds. So you get all four, but I need you to list all four. And so the tertiary level is one polypeptide, fully folded, 3D shape, all four bonds. The quaternary level, you have fully folded polypeptides. They have 3D shape, but it's more than one polypeptide. So it's a bunch of these folded together and joined together. A bunch of these polypeptides. Some proteins, like insulin, just need one polypeptide to be functional. They're functional at the tertiary level. But many, one of my favorite proteins is hemoglobin. It carries oxygen in blood. It's only functional at the quaternary level. So different proteins are functional at different levels. They could be functional, if you're making a general statement, proteins could be functional at tertiary. If they're not functional at tertiary, they are functional at quaternary level. So here's an image of the primary level. Again, it's just the order of amino acids. It's pretty linear, it's not folded. The secondary level, here's an alpha helix, and here's a beta sheet. And you get those by local folding, partial folding, and usually with hydrogen bonds. The tertiary structure, you get one polypeptide fully folded, and you can see helices and beta sheets in here with some what we call loops, which are just part of the amino acid chain that's kind of free. They're not in a helix or a sheet. They're kind of their own little segment. And then this, I do believe, is hemoglobin. So this is the quaternary level. You have 2 alpha polypeptides and 2 beta polypeptides. And they're all joined together to make this large structure. So those are the 4 levels of protein structure. And that's going to be one of the big pieces of the protein section, as well as the structure of an amino acid and knowing what a peptide bond is. Some proteins are chaperone proteins, and they help proteins fold properly. And we found out that some of them may be pretty important because if they don't function, proteins will misfold. And when they misfold, they typically get sticky, and they stick together and make these big complexes that kind of take over the cell and usually end up killing the cell. And that's an important pathology in Alzheimer's and cystic fibrosis. Prions are some creepy zombie proteins. They are also misfolded, but in a way that is contagious. So this misfolded protein bumps up against a protein that's folded normally, and it sort of scrambles it and rearranges it and until it's misfolded. And mad cow's disease is its disease follows this pattern. And there's some other encephalitis. There we go. That follow that pattern